What are plant hormones? According to plant physiologists like Frank B. Salisbury, plant hormones or phytohormones are chemical messengers produced naturally by plants in extremely low concentrations. These organic signal molecules regulate virtually all aspects of a plant's life, controlling the plant's growth, development, and response to its environment. In the next following videos, we are going to talk about the hormones, its classification, and key functions.
Auxins are concentrated in a growing tip. Removing a tip, a practice called pruning. Stop auxin dominance. Tilling the plants to get bush, which is important for vegetables and fruit production.
Cytokinins are the cell multiplication hormones. They work against auxins to control the balance of shoot and root growth. Agriculturists use them to promote lateral branching and keep harvested crops fresh longer.
Gibberellins are essential for turning a seed's potential into a reality. They initiate germination and are often applied externally to crops like mung beans to increase stem length, leading to larger and better spaced clusters.
AVE is the plant surveillance. It stops growth and signal for dormancy in seeds and plants. Most importantly, it's the primary commander in a biotech stress response.
Ethylene is the only gaseous hormone and it is the hormone of pollination. It triggers fruit ripening and senescence or aging. This is a vital biological process for managing harvest and post-harvest product.
And now let us summarize the biochemical technicalities of the 5 classic phytohormones. First, let us begin with auxin. Auxin is an indole 3-acetic acid synthesized from the amino acid tryptophan. Its mechanism is that auxin binds to its receptor and targets repressor proteins for destruction via the cell's protein disposal system. The destruction of these repressors releases other proteins called auxin response factors, which then switch on the genes needed for cell elongation and growth. The second one is cytokinin. Its active form is zeatin. Chemically, it is a derivative of the base adenine. Cytokinins bind to receptors embedded in the cell membranes. This binding starts a cascade where phosphate group is transferred from one protein to the next. The final phosphorylated protein activates genes for cell division or cytokinesis, lateral shoot development, and delay leaf aging. The third one, gibberellin, its active forms are GA1 and GA3. Gibberellin binds to its receptor, which then attaches to and targets inhibitor proteins known as delta proteins for destruction by the protein disposal system. Since delta proteins normally suppress growth, their removal allows the cell to activate genes to promote rapid stem elongation and seed germination. For the fourth one, we have abscisic acid. It is synthesized from carotenoid molecules. ABA binds to its receptor, PYR or PYL proteins, and this complex then immediately shuts down a key enzyme known as protein phosphatase. Shutting down the protein phosphatase releases another key enzyme, a kinase, to become active. These active enzymes control the ion channels in the guard cells, causing the stomata to close and save water, which is the primary stress response. And for the last one is ethylene. It is a simple volatile gas. In a normal non-stressed plant, the ethylene receptor is active. and continuously shuts off the ethylene response. When the ethylene gas is present, it binds to the receptor and inactivates it. Inactivation of the receptor removes the repression, allowing the signal to proceed and activate genes for fruit ripening, leaf shedding, and aging. In a changing climate, plants rarely face just one threat. They need a complex communication network. How do they biochemically manage that? By utilizing the classic hormones. And in this segment, also introduce more signal molecules that fit the definition of a hormone. Chemicals that, even in low concentrations, regulate growth, development, and stress responses. The ABA from before is the crisis manager. It triggers stomatal closure but also induces protective changes at the cell level, like producing osmoprotectants to save water inside the cell. We use this knowledge to breed plants that are more sensitive to ABA signals.
Defense against pests and disease is split. Jasmonates fight chewers-like insect. Salicylic acid fights infection. The practical application for us is chemical primering using non-toxic biostimulants to wake up the GA pathway before the pest arrives, leading to a faster, stronger, and more sustainable defense.
We've seen that plants are not passive, they are sophisticated biochemical engineers. Every growth, every fruit, and every defense mechanism is governed by hormone signaling and molecular processes.
The future of agriculture lies in moving beyond simple chemical application to understanding the chemistry of the crop itself.
As future agriculturists, our role is to leverage this biochemistry.
Whether it's using genetic technology to make the ABE stoma response more efficient, or using biostimulants to trigger the jasmonate pathway before a pest arrives.
We are using fundamental biochemical knowledge to create crops that are healthy, more productive, less resilient on external chemicals, and resilient to the challenge of climate change.